Species-specific impact of microplastic on corals' physiology. We live in an age with lots of waste. Between 60 and 80% of everything we throw away is plastic, and about 10% of the annual plastic production enters the sea. This has impacts on marine wildlife, which leads to the death of many species. Without using a microscope, we can only identify the big pieces of plastic, but responsible for the death of animals and plants are microplastics. These are fragments smaller than 5 millimeters, which can be subdivided in two different forms: fibers and spheres. The main reason why microplastic is so dangerous to corals is that these animals are not able to distinguish the fragments from their usual food because of its similar size. There are already some studies about the correlation between corals and microplastics, but none of them uses the concentration of microplastic which corresponds to the real situation in the sea. For this study, scientists made an experiment to find out about the impacts which microplastic has according to the current topic of climate change. To answer the questions, scientists set up 4 aquariums, all of them with identical lights and pumps. The first tank included 1 piece from each of the 2 coral species. To simulate reality, scientists calculated the concentration of microplastic in the sea and added it to the water. They also kept it at a constant temperature of 26 degrees. In the second tank, they put the same as in the first, with the only difference that it has a temperature elevation from 26 degrees to 32 degrees, 0.5 degrees every 2 days. The other 2 tanks were copies from the first 2, just without microplastic. They were only used as a reference to the others. After 12 days, all the collected data from the photochemical efficiency was put in the table. Between the 2 species, we see that Acropora reacts in general more significantly to microplastic than Ceratopora. It is also evident that an increase in temperature has an effect on the photochemical efficiency of both of the coral species. If we only look at the simple experiment without temperature increase, we see that Acropora reacts significantly stronger to microplastics than Ceratopora. In detail, fiber exposure causes more damage to the photochemical efficiency of Acropora than spheres. With Ceratopora, we see the opposite of that. The increase of temperature causes in all cases a higher photochemical efficiency than the control tanks. According to the physiological stress, there was observed nothing special after the temperature elevation.